if a pathogen invades beyond your first and second line defenses, there are different chemicals and components in the cells that are able to help out, able to give a further defensive mechanism. Now you can see here there's a whole list of what these are. From phagocytosis, there's many different cells that are phagocytes. You have natural killer cells. They're going to do what they say, literally kill the cell that's infected. You've got interferons and opsonization. We'll get to more of those later. But like the name implies, interferon is going to interfere with replication of viruses. You have complement proteins, inflammatory response, fever. All of these are important mechanisms in preventing further spread or trying to get rid of what's already there. The inflammatory response, we've mentioned it before, because inflammation helps to kind of cordon off an area and allows more cells to come in as well. Fever, well, I'm sure everyone's had a fever. Body temperature rises. That increased temperature helps to increase the activity of your body cells. So let's look at phagocytosis first, these phagocytic cells. Now, when we're back in the blood chapter, we talked about plenty of the different white blood cells and what their roles are. But in general, phagocytosis, you're going to have the cell, which in here is kind of purplish color, needs to find a pathogen, has to be able to identify it. But it might not be a pathogen, it might just be cell debris, an old dying cell, whatever it's trying to destroy. But for our purposes, we're going to focus more on pathogens because this is the immunity chapter. So they have these pseudopods that kind of grow out. They're not really normally there, but they're just growing out around the pathogen. You can see how you have this piece here and this piece here are kind of coming off the cell surface to create a capsule around the actual pathogen. Then once they fuse, they pull it inside. You're going to have this lysosome here joining with that capsule, with that vesicle. By allowing it to join, the lysosome, those enzymes, can start to break down and degrade the pathogen, destroying it until it's non-functional. Once you're down to the basic building blocks, it's not going to do any harm, so you can simply get rid of it, dump it out of the cell through exocytosis. This way, that phagocytic cell can just keep on going. Find another pathogen, destroy it. Find another pathogen, destroy it. So here you have a micrograph. You can see the actual macrophage is right in the middle. Now the green on the outside here and here, those are going to be the pathogens. Those in this case are bacteria. They're reaching out, those pseudopods are reaching out to engulf it and they'll pull it back in. So once that's pulled into the actual macrophage, then the lysosomes will destroy it and that has been neutralized. So we're looking at destruction. That's the key thing. Destroy the pathogen. Destroy the foreign particle. So a couple different ways of looking at here is acidification. Because a lot of times bacteria aren't very functional in very acidic environments. It's one of the benefits of your stomach, for example. But the cells can create an acidic environment. And of course, we mentioned lysozymes. So you're looking at the enzymes in those lysosomes. So different ways that you can have destruction of pathogens via phagocytosis. Now we have this thing called a respiratory burst. Now in the case of a respiratory burst, you're releasing these free radicals. Well, the free radicals can kill cells. Just gotta be careful because free radicals in your body can kill many more than just pathogen cells. So this isn't always a go-to mechanism, but one way to get rid of a lot of cells. And there's more enzymes, of course. Your body has more enzymes than what's just in the lysosomes. So you have other cells with other enzymes that could be specific to different things. And then your neutrophils have these things called defensins. Again, more components, more chemicals that are meant to destroy pathogens. So looking at the macrophages, they're coming from the monocytes, if you remember. So now, that's not the only way that you have phagocytic cells, but this is how you get those macrophages, coming from the monocytes. And that's primarily the most advantageous, the most destructive of your phagocytic cells. But you have two different types. You have free macrophages. These ones are simply floating throughout your blood looking for anything that shouldn't be there. Whether it's a pathogen, an old dying cell, cell fragment, dirt or debris, or whatever. They're simply free-flowing and looking to destroy and get rid of what should not be there. It's a way of trying to clean things up. But then you also have fixed macrophages. Now these ones are anchored in place. They will not move. They're watching what travels beyond or past them. Examples here. You have these comfort cells in the liver. Well, that makes sense. The liver cleans your blood, so blood's constantly flowing through the liver. These cells are monitoring that blood. You have microglia. Hopefully that rings a bell from AP1. Microglia are in your central nervous system. They're anchored in place, and they're watching the fluid pass by in your central nervous system. So both free and fixed macrophages. Both do the same thing, trying to get rid of foreign particles, whether it's pathogens, dying cells, or simply dirt and debris. The next one I mentioned before in that list were the natural killer cells, or NK cells. Now these natural killer cells, interestingly enough, are not phagocytotic. They're not going to simply go through the process of phagocytosis. Instead, these are large granule lymphocytes. They're pretty much targeting when you're lacking or you don't have the self-antigen. Now, almost every cell in your body has that self-antigen. It's how you know which are your cells versus which ones aren't your cells. The natural killer cells look for cells that don't have that self-antigen. If you're saying I mentioned almost all, that's true, almost. There are some that don't have that, and they have to be kept separate from your immune system. Otherwise, your immune system would destroy them, for example. So these ones are not specific. It doesn't have to be a particular bacteria or a particular virus. As long as it doesn't have the self-antigen, it gets destroyed by the natural killer cells, or marked for destruction. Because what's happening here is, it's looking more to induce apoptosis. Now, apoptosis is cell destruction. Kind of think almost as cell suicide, destroys itself. This is great for cancer cells and virus cells. And when I say virus cells, I mean the cells that are containing viruses within, because viruses have to replicate inside of your own body cells. So what's happening here then is, these natural killer cells can recognize those cells and say, hey, trigger the apoptosis reaction. Now that gets tricky though sometimes, because in cancer cells, some cancer cells have the self-antigen. So it's not a foolproof catching every cell, but trying to get as many as it can recognize and cause destruction of. Then we have these interferons. Now interferons, as I mentioned before in that first slide, kind of interfere with the virus replication cycle. What they're going to do is they actually are going to be antivirals that can activate an early protein production. The way that works is, a virus invades a cell. That cell can produce these interferons. The interferons go out to the surrounding cells of that infected viral cell. They tell those cells to make antiviral proteins, which makes it harder for the virus to get into those cells and replicate. So it's a way of trying to not necessarily protect itself, but protect the cells around it, more for the greater good. Also, can deal with activating macrophages and help with the natural killer cells. You're saying, why do you need those? Or stopping it by telling it not to reproduce. Yes, the local environment, but those viruses can spread beyond the local environment. So you're getting the immune system up and ready to function, ready to go at further distances from that localized area. Now, there are genetically engineered IFNs. These interferons can be genetically made. The benefits here, hepatitis, genital warts, those can have IFNs that are genetically made to help and try and stop or prevent those. Now, with viruses, you can never fully get rid of them. Just try having them go into remission. So what they're doing is trying to make sure they're not expressing those virus particles anymore. The viruses can't simply reproduce more because the cells are resistant to them. There are some multiple sclerosis treatments that are using genetically modified or genetically engineered interferons, trying to help those patients not necessarily regress and go back to the way they were, but prevent further progression of the actual treatment there. So it's trying to prevent multiple sclerosis from continuing on the process, trying to see if they can't help those individuals at that point. So there are a bit of different genetically modified ones and research going into these to seeing other ways that can be beneficial to help treat patients and treat individuals that in the past couldn't be really treated very well. So these interferons. So kind of a visual way of what we're just talking about. Over here, you're looking at, first of all, the virus enters the cell. When that virus enters the cell, it's then going to cause the DNA to basically say, hey, you know what? A virus just invaded. We need to make sure that we now produce these interferons, these particles. Those interferons are received by cells that are close by, and it triggers those cells produce this blocking agent. So therefore, when the new virus tries to come in, that virus comes in and immediately gets blocked and stopped. So it's a way of, again, protecting the surrounding area, the immediate cells around that particular infected cell. It means that particular infected cell still can produce virus particles, and that infected cell can still send those particles out further away. But at least the closer cells can't get infected right away. So it slows the process down. All right, opsonization. Now, the case of opsonization is marking a pathogen for phagocytosis. Pretty much what that means is this is saying, hey, tag, you're done, and allowing someone else to find that and destroy it. So it's not going to actually destroy it itself. It's just putting a big red X on the back of that pathogen. So the phagocytic cells can then find it and destroy it easier. So opsonization is basically taking and having antibodies or even antimicrobial proteins binding to this pathogen that will mark it for destruction. So it's a way of trying to help your body identify what shouldn't be there. So a couple of different ways your body is using to fight and get rid of infections inside your cells.